Okay, so we are done with DNA. And now it's time to move from DNA to RNA in a process called transcription. What is RNA? RNA stands for ribonucleic acid, very similar to the name of DNA. And we often think about RNA as the intermediate or messenger. It's not really the set of instructions like a book in terms of storage, but it's really getting in between the book and doing the action. So if we think of the DNA as like a book of instructions, maybe the RNA is you, the person reading the instructions, so you know what to do with those instructions. It's an intermediate. You haven't done anything with the instructions yet, but you've read them and you know what they are. RNA is how DNA is read, and it's the intermediate before converting those instructions to the thing that can carry out the instructions, the molecular tool, protein. In reality, RNA is more than just this intermediate between DNA and protein. It can do chemistry, it can do some of the functions that protein can do. But for the sake of this class, we'll think of RNA as a kind of intermediate message. What is RNA? Well, let's compare the structures on the right. We learned how DNA has 2 strands. Compared to DNA, RNA only has a single strand. Similar to DNA, RNA has 1, 2, 3, 4 bases in its code. But you'll see, rather than thymine, RNA has a similar base called uracil, but it's slightly different. And so this is one way to easily distinguish between DNA and RNA, is that DNA has the T, whereas RNA has the U. Because RNA is single strand, we don't necessarily have the type of base pairing that you see with DNA, where A is always with T and C is always with G. RNA can fold in on itself to perform base pairing, but DNA is base paired basically throughout, whereas RNA might fold to a little bit of base pairing and not do base pairing somewhere else. RNA is an exact copy of the original DNA message, right? So if the sequence of DNA is C, G, A, T, the sequence of RNA is C, G, A, U, because we've replaced T with U. Now, when we've converted DNA into RNA, we have this intermediate messenger. And we need this messenger to go ahead and perform some action to carry out the instruction. And so we need to convert this RNA into protein through a process called translation. Now, you've likely heard of proteins. You know you need to eat protein in your diet, for example. Proteins are often thought of as the tools of the cell, right? So let's say that the book of DNA instructions was read by you, and then you reading the instructions and knowing the instructions, that's the intermediate message. Well, the protein would be like you actually doing something with those instructions. Maybe you build a chair. That is the action, right? The chair being built is the action, so we can think about that as the protein, for example. Proteins perform all sorts of functions. They come in all shapes and sizes, a variety of tools for the cell. They can perform chemical reactions, they can make bonds and break bonds, like we've learned about. We call these enzymes. But those proteins can act like small machines, and they can do physical work, like they can move in a circle, or they can have like a walking motion where they move from one place to the other in a cell. Proteins can also talk to each other, so proteins can come together and kind of pass a message along within the cell, and we call those receptors and signaling molecules. We will talk a lot about receptors and signaling molecules when we start to learn how drugs work in this class. Now, proteins are very different than both DNA and RNA. The structure of DNA and RNA is very similar. Proteins are different. Proteins are made up of very small chemicals called amino acids. And there can be really tens of amino acids to thousands of amino acids that make up a protein. So they can be very, very small proteins like the peptide insulin in your body, or they can be very, very large proteins like the protein, I believe the one that's here on your screen is hemoglobin, that makes your blood cells red. There are 20 total amino acids, and their order matters. The order of amino acids for each protein is unique, as is the number of amino acids in each protein. And the order of amino acids came from the order of A, U, C, and G in RNA. So the RNA is literally read out to make this protein, and the letters and their sequence in RNA tells the protein what sequence of amino acids it needs to have. Now, while in this class, we're going to be focusing a lot on receptor targets of drugs, it's also important to know that enzymes, so proteins that do some type of chemical reaction, those are also a major target for drugs. And it's very likely that drugs that all of us have taken targeted enzymes. And that wraps up the central dogma, folks. What did we learn? We learned that DNA are a set of instructions. DNA can replicate itself when we have to make more cells and divide our DNA from one cell into two. We learned that DNA is turned into RNA through a process called transcription, and RNA is this intermediate messenger. And that message is read out to make the protein tool in a process called translation. And that is the central dogma. I'll leave you with this cat meme, the cat's dogma. See you in class.